I was asked to talk about difficulties and barriers in developing RSV polymerase inhibitors, and I'm going to mention what the barriers are, but I'm really going to focus most of the talk on what we can do in terms of studying RSV polymerase and the assays that are in place for that. So at the bottom here, I've just listed the companies with which from whom we've received support over the last few years for our research. Hang on. Okay, here we go. Excuse me, I've got a bad cold. I have a respiratory infection, so I apologize, I'm losing my voice. So I thought I would skip the sort of background on RSV impact on public health, because I think a lot of people know it, and there's other speakers who are going to cover that later today. So I'm going to just dive into the molecular virology. And here what you can see is a very simplified schematic of the RSV replication cycle. And if we start up here, here's the virus particle, and that begins by releasing the viral genome into the cytoplasm of a host cell. And the RSV genome is a single strand of negative sense RNA. And so associated with the genome is the viral polymerase, shown here in orange. And during the viral replication cycle, that polymerase has two roles. It transcribes the genome to produce capped and polyadenylated messenger RNAs that correspond to each of the genes, and it also replicates that genome so that there are progeny genomes that can be packaged to make new virus particles. So obviously the RSV polymerase is absolutely essential for the virus to multiply and replicate, and so and for that reason it's a good target. So how does the polymerase actually do these tasks? This is a schematic of the RSV genome here, and each of the genes is illustrated with a blue box. And what happens during transcription is that the polymerase will come and bind to this genome at this 3' leader region here. So this is a promoter region that recruits the polymerase, and then the polymerase can move along the genome and respond to signals that lie at the beginning and at the end of each of the genes. So these signals will direct the polymerase to initiate RNA synthesis, cap the RNA, methylate the cap, and then when the polymerase gets to the end of the gene, it polyadenylates the RNA and releases it, but can then reinitiate at the next start signal. So in this way, it's able to make all these different messenger RNAs. What happens during replication is that, again, the polymerase is going to bind to this promoter region, but in this case, it disregards all those signals and synthesizes the full-length complement, this antigenome, which then acts as a template for genome synthesis. So one of the differences between transcription and replication is that the replication products get encapsulated with a protein called nuclear protein or N protein as they're synthesized. So they form so if you can imagine it like beads on a string, it forms this helical ribonuclear protein structure, which I've illustrated here. Okay, so I think you can get from this that the polymerase is quite complex. It does a lot of different tasks. And we have no structure for the RSV polymerase. This is the structure of the VSV polymerase that Matthias just referred to, which is the closest thing that we have to a full structure for a polymerase of the non-segmented negative strand RNA viruses. And so the polymerase is basically 3 enzymes in one protein. There's the RDRP domain, shown here in blue. The polymerase is able to cap the RNA, so it has a capping domain that is quite distinct from any other cellular capping enzyme. And then it has a methyltransferase that allows the caps to get methylated as well. And so why is this challenging to study? Well, partly because it's a very large and complex enzyme. And the RSV polymerase is over 2,000 amino acids long. It's 250 kilodaltons, and that's for the large polymerase subunit that contains those enzymatic domains, this bit I've just shown you. But it also requires a cofactor, a phosphoprotein P, in order to be expressed. And that P protein, although it's small, is a little tricky to work with because it's quite sticky, so it's quite hard to get highly purified polymerase preparations. And so I think for these reasons, the field is lagging behind, and we don't, as I said, we don't have a high-resolution structure. The only thing that we've got really close in terms of really high resolution is a structure of the C-terminal fragment of the human metapneumovirus polymerase, the methyltransferase domain. So that's a closely related polymerase. So that's one challenge in studying polymerase activities and coming up with polymerase inhibitors. The other challenge in studying the polymerase is that the template isn't naked RNA, it's encapsulated. And so what this means is that in its natural context, the polymerase never sees large stretches of naked RNA, and it never has to deal with RNA secondary structure. And so we're very limited in what templates we can give a purified polymerase. And trying to reconstitute this encapsulated RNA in vitro has so far proven to be elusive, at least for long lengths of RNA. So without a structure and without and with challenges in sort of reconstituting polymerase activity in vitro, we are kind of challenged in developing inhibitors, but there is quite a lot that we can do nonetheless. So I'm going to focus now on the positives. And basically, what we can do is if people can come up, well, I'm saying what I can do, or people can do, if people come to us with an inhibitor that they think is a polymerase inhibitor, we do actually have a number of studies that we can, assays that we can implement to determine mechanism of action of those inhibitors. And I'm going to go through briefly some of those assays now. So there are ways that people can screen for inhibitors. For example, there is a replicon, cell-based replicon system for RSV. And if there are hits in that system, then that's a strong indication that the compound is inhibiting some part of the RSV transcription replication machinery. And then with that information in hand, we can then go on and apply a combination of cell-based assays, which we call mini-genome assays, and then biochemical assays with a purified enzyme to study the mechanism of inhibition. So the mini-genome assay was developed about 2 decades ago by Peter Collins's group, and it's illustrated here. So this is a little different from the HCV replicon. It's a transient expression system. And basically what we have is the RSV genome from which the viral genes have been removed and replaced with reporter genes, but maintaining the cis-acting elements that enable transcription and replication to occur, so the ends of the genome and the gene junction. And if that mini-genome is expressed in cells together with the viral polymerase, the nucleoprotein that can wrap it up and encapsulate it, and then a transcription elongation factor protein, M21, then it gets wrapped up non-specifically by N protein, it's used as a template, and you can get messenger RNAs and a little antigenome. And then this antigenome can then act as a template to make more mini-genome. So what we do in our lab is to modify this actually, and we limit it to a single cycle of replication. So what I mean by this is that we make a mutation here in this trailer region here, which basically inactivates the promoter at the 3' end of the antigenome. And what that does is it means that we can look at transcription and replication as independent events. And so if there is an inhibitor, we can ask, is it blocking transcription and replication, or just one of those processes or the other? We can also, in this system, it's a very robust system, and we can even see abortive RNA synthesis products. So we're able to distinguish between initiation and elongation as well. We're also able to analyze the 5' ends of these messenger RNAs by primer extension and enzyme digests. And we can determine then if there is a cap at the end. We've also now determined that we can look at 2' methylation of those RNAs. And we can also use assays to determine if the RNA has become encapsulated or not. So we have this whole kind of menu of different assays that we can use to look at RSV polymerase activity in a cell-based system. Then we also have an in vitro assay for looking at RSV RNA synthesis activities. So in this case, what we do is we purify the polymerase using a baculovirus. We express it using a baculovirus vector and then purify it. And as I said, it's a complex of the L and the P proteins. Excuse me. And then we can take that and incubate it with a promoter RNA, a short little promoter sequence, and radioactive NTP and other NTPs, and we'll get RNA synthesis products, as you can see here. And what we've done over the last few years since we developed that assay is to really kind of investigate polymerase activities using both cell-based assays, looking at virus, and and then looking at this in vitro system. And what's quite gratifying is that everything that we look at in cells is also matched by what we see in vitro too. So here are a few examples. So we know that the RSV polymerase actually initiates at 2 positions in the promoter. So it will initiate replication opposite position 1U, and it initiates transcription opposite position 3C. And you can see that here. So this is looking in RSV-infected cells at the 2 initiation sites. And when we look in our in vitro assay, again, using a promoter sequence and look to see where the polymerase initiates, we can see that, again, it initiates at positions 1 and position 3. We've also shown that the RSV polymerase can become preloaded, or I should say, can select the initiating NTPs independently of the template sequence. So it has a natural affinity for the initiating NTPs. And we've shown that in a cell-based assay, and we've shown that also in this in vitro assay too. Another thing that the RSV polymerase does, which is a slightly odd activity, we don't know why it does it, but when it encounters the 3' end of the answer genome, if that answer genome 3' end is naked RNA, the polymerase can fold that into a secondary structure and add 3 nucleotides in a templated fashion. So here it folds it like this and then adds on a G, a U, and a C. And this is sequence analysis of RSV antigenome RNA from infected cells, and you can see that additional G, GU or GUC here, and we see this also in the in vitro assay. So this is putting in that, that 3' end of the antigenome. It will act as a promoter, and we get RNA synthesis, but then we also see products longer than the input template, these 3 bands up here, and when we determined the sequence of that, we determined it was GUC, just like we saw in cells. So this is quite gratifying that this polymerase, this purified polymerase, although it's a very artificial system, it does behave very similarly to what the polymerase does in cells. And so with that, we've gone on to do some inhibitor studies, and I'm going to breeze through these very quickly because they're all published, of how different inhibitors function. So we've looked at AZ27, BID, and GS5734. So this is looking at AZ27. AZ27, we showed with our mini genome system, inhibits transcription, you can see that here, and it also inhibits replication, and it inhibits them both to about the same extent. We were able to show it inhibited a very early step in both of these processes, and then we went to our in vitro assay and then showed that it actually inhibits the formation of the first dinucleotide, so this 2 nucleotide product is inhibited by AZ27. Looking at BID, BID was a compound developed by Boehringer Ingelheim, and it was known to be a capping inhibitor. And we confirmed it's a capping inhibitor, but we showed it had another activity as well. So in our in vitro assay and in cells, we know that the polymerase, when it initiates position 3 of the template, it normally just releases the RNA after 25 nucleotides. So you can see that here. This is what we normally see. And in the presence of BID, we saw that actually the RNA was elongated further so that it got to the end of the template. So BID caused the polymerase to be more processive, it actually increased elongation. And we showed that that also happened in the mini genome system. So as I said, transcription initiates at position 3, and the first thing the polymerase makes is a short 25 nucleotide transcript. And then it reinitiates RNA synthesis at the start of the first gene. And what we found was that in the presence of BID, this transcript got elongated, and the polymerase then couldn't initiate so efficiently here. And you can see this here in the primer extension analysis. And then finally, the GS5734 triphosphate, we looked at that in the in vitro assay, and if you just look here at the side with minus ATP. So in the absence of ATP, we would get a 3 nucleotide product, which you can see here. And in the presence of the GS compounds, but no ATP, we can get incorporation of the compound in place of ATP, but only for a certain number of insertions. And then after that, the polymerase fell off, and you compare this to native ATP over here. And so RSV polymerase can incorporate multiple copies of GS5734 triphosphate, but then it terminates. So this is similar to what Matthias was saying happens with Ebola virus. Okay, so in the last couple of minutes, I'm just going to talk about what we're going to do, or what we're trying to do in the future. So we have this in vitro assay for the polymerase, where the polymerase seems to behave very well. And so we thought, well, it would be very nice if we could develop that as a screening tool, so that you could look for things that are specific polymerase inhibitors. And obviously, to do that, we would need to get away from using radioactivity, which is my major cost on my grants is radioactivity. This is the design that we've been testing out. What we thought we would try and do is set up an assay where we had the polymerase use a primer instead of de novo initiation, and a primer that would have a modification on the end. And we know from Jerome Duval's work from Alios Biopharma that the polymerase will tolerate a primer, but we didn't know if it would tolerate a primer with a modification at the end. So if we can have a modification at the end, we could have something like biotin or a fret floor or something like that to act as something that can anchor it or act as a part of a signal. And then to ensure that our primer was being used and not de novo initiation, we incubate it with a non-specific template, so no promoter sequence in here. And NTPs, including an NTP with a modification on it. So again, something like a fluorescent NTP or something that could be used as a fret partner that then we could detect. So the stage that I'm showing you now, unpublished data, and the stage that we've got to is to just kind of test out what the polymerase will tolerate. And it's actually reasonably tolerant. I'm quite surprised. So this is looking at primers with 5' modifications. We looked at biotin, tetroxygenin, and Cy5. And here it is with just a normal 5' hydroxyl primer, and that's our positive control. And with the modifications of biotin or tetroxygenin, the polymerase can still use those as primers. Unfortunately, with Cy5, it doesn't work so well. Probably this is too bulky and not getting out of the exit channel. And then we looked at different NTP analogues, some ATP, GTP, and UTP analogues. And again, you can see actually the polymerase is reasonably tolerant. It doesn't incorporate all of them. It certainly doesn't incorporate them as well as the native NTP. They will incorporate a number of different analogues that are fluorescent or that could be modified by a click chemistry reaction, such as ethanol UTP. And so to summarize, the RSV polymerase is a promising target. It's essential for the virus to replicate, and it's basically 3 enzymes in one protein to target. But the challenges lie in the size and the complexity of that polymerase and the difficulties in purifying it to homogeneity. And then also the nature of the template, and I think that's a very difficult aspect of it to overcome. That's going to be very difficult to reconstitute in vitro. But as I said, we can do a lot of things using cell-based assays and then complement those with biochemical assays to determine the mechanism of inhibition of any inhibitors. And now our future work is focusing on trying to develop that in vitro system so that we can try and get something that would be useful as a screening tool. And with that, I'm going to just acknowledge the people. This is my lab currently. The people in bold are the people who contributed work to the talk. And then these are the industry collaborators and the funding sources. Thank you.